Global and local excitation and inhibition shape the dynamics of the corticostriatal thalamocortical or CSTC pathway. The seat of the CSTC pathway is the basal ganglia. What is the basal ganglia? The basal ganglia are a group of structures found deep within the cerebral hemispheres. The structures generally included in the basal ganglia are the caudate and putamen, striatum, and globus pallidus in the cerebrum, the substantia nigra in the midbrain, and the subthalamic nucleus in the diencephalon. The nuclei of the basal ganglia are interlinked via two CSTC pathways, the direct and the indirect. In the direct pathway, excitatory neurons from cortex end in striatum. Inhibitory connections from striatum in turn reach the globus pallidus internal and the substantia nigra pars reticularis, which inhibit the thalamus. When the striatum inhibits globus pallidus internal and substantia nigra pars reticularis, their activity of inhibiting thalamus is reduced and it can excite cortex. The indirect pathway acts to inhibit the cortex. Inhibitory connections from striatum end in globus pallidus external and subthalamic nucleus, which excites the substantia nigra pars reticularis, enabling it to inhibit the thalamus, which is in turn unable to excite the cortex. The medium spiny neurons of striatum in the direct pathway contain D1 receptors, and those in the indirect pathway contain D2 receptors. Dopamine excites the direct and inhibits the indirect pathway. In this paper, activity in the above discussed 7 nodes of the CSTC pathway were modeled by the Wilson-Cowan model. Matkant package was used to study bifurcation diagrams and locate attractors while varying global and local excitation and inhibition strength in the pathway. Plots showed major influence of local strength around D1 and D2 MSNs on hyperactivity, which is implicated in neurobiological disorders like OCD and Tourette's.